Hi everyone, welcome to COVID Part 2. This is Dr. Collins Jones with the Biotechnology Program here at Montgomery College. I hope that you will find today's presentation interesting and hopefully useful as well. Today we're going to cover a really quick review of COVID-19, a quick review of how COVID spreads, and then spend a few minutes on COVID statistics, look at COVID diagnostic tests that are on the market or in development. In the May presentation, we explained how those tests work. So we're just going to describe some of the current tests and what's going on with those. Look at COVID therapies, ones that are currently working, ones that are in the pipeline. And then we will discuss COVID vaccines. We'll start by explaining how a vaccine works. Then we'll go into how vaccines are developed and manufactured. And we'll end up by looking at a couple of vaccine candidates. Let's get started. If you attended the presentation back in May, you might recall that this is the model that I used to represent COVID-19, the COVID virus. Right? It was a fairly simple model, but hey, we're only 5 months into the pandemic, and scientists were really racing to understand more of the details of the structure of the virus, the nature of the virus, and so on. Well, here we are 10 months into the virus essentially, and things have changed. We now have a lot more information about the structural detail of the virus that helps us to understand how the virus works. And by understanding the structural and functional details of the virus, it helps us to develop diagnostics, therapeutics, and vaccines. Today, I just want to quickly focus on two aspects of the virus. The first is single-stranded RNA. RNA is the genetic material that contains the instructions to make new virus. So recall, once a virus enters into the cell, its RNA is released, it begins to make copies of itself. So the RNA contains the directions to make all of these proteins that are present inside and outside of the virus. So there's our little piece of RNA. So we use this single-stranded RNA, diagnostically, right? This RNA is unique to the coronavirus. So we use that as a way to detect the presence or absence of the virus. We try to see if corona RNA is present or absent. In addition, if we can block the replication of the RNA, if the virus can't make new copies of its RNA, then there won't be any genetic information to incorporate into new viral particles. So therapeutically, people are looking at molecules that may prevent the replication of this single-stranded RNA. The other molecule that we focus on is the spike protein. Right? So the spike protein is a surface protein on the virus. It's on the outside surface of the virus, and the spike protein interacts with the ACE2 receptor on your lung cells, on the cells lining your throat, and when the spike protein binds to the ACE2 protein, that allows the virus to be internalized inside the cell, where it can now come apart, expose its RNA, and begin to make copies of itself by making all of these protein molecules. So there's a lot of focus on the spike protein, particularly when it comes to vaccine development, because when the spike protein interacts with the ACE2 receptor, again, that is how the virus can infect a cell. And so what we want to do is prevent that interaction, right? And so if the virus cannot recognize and bind to the ACE2 receptor, then it can't infect a cell. If the virus can't infect a cell, it can't replicate. It just floats around in your blood, and eventually your immune cells will detect it and kill it, and just totally eliminate it. With that quick review, let's talk a little bit again about how COVID spreads, right? Again, the main transmission route for COVID is believed to be close contact, person to person, via respiratory droplets, right? Little droplets of liquid, of fluid that come out of your mouth, that come out of your nose, all right? Talking, coughing, sneezing, singing, and breathing generates these droplets. The louder you talk, the bigger the droplets, the more droplets that come out of your mouth and potentially out of your nose and begin to float through the air land on other people, land in other people's noses, land in other people's throats. And if any of those droplets have COVID in them, then you've now infected those people. So this is a picture I pulled off of the internet outside of a bar in New York City, right? And you can see that lots of people here are protecting their chins and necks from exposure to COVID, but not their nose and mouth, all right? So proper mask wearing, we'll emphasize on the next slide, is really important. I have seen some studies in the last month or two where some scientists claim that the micro droplets, not the big ones, but the really small droplets, may actually travel more than 10 feet. And because they are small, these micro droplets can remain airborne for minutes to potentially hours, depending on the environment. Turbulence from HVAC systems may keep the droplets aloft, and it may increase the distance that they travel. So, part of restaurant transmission is you're sitting in a small space, you're close to each other, and the air conditioning or the heating is on, and it's basically creating wind that's circulating everything all around the restaurant, exposing everybody to everything, even if you're 10 feet away. But on the other hand, in some cases, especially if the door open a little bit or a window cracked, this may actually serve to disperse, to dilute these micro droplets containing the virus. And again, part of whether or not you're going to get really sick is the dose or the burden of virus that you inhale, that you're exposed to. So the less virus you're exposed to, the less likely you are to become sick. One of the things to think about that I'm very aware of, especially with the holidays coming up and kids coming home, is the bubble that you think you have is not really the bubble that you have. So here you are in your own little bubble. These are all the people you're interacting with, okay, on a regular basis. None of these people have COVID. Somebody over here in their own little bubble has been exposed to COVID, and they may now be a carrier. They may be able to transmit it. And if they transmit it to this person who now interacts with you, the virus that was over here originally outside of your bubble has now entered into your bubble, and you may now be exposed, and you're going to expose all these people who in turn will expose these people. So this is, one of the ways that while you think you're safe, you may not actually be safe. So we just sort of have to be aware of that. All right. Okay. Now, a little bit of statistics. Okay. So Kevin Long, you can do a much better job of pulling things up. I've seen what you've done helping us with some of the grants we're writing now. Just amazing. So this is my little simple version. Don't worry, your job is safe. Okay. So one of the things we want to see, I picked this up just a couple of days ago. It's November 4th as I record this. But coronavirus around the world, there are almost 50 million confirmed cases worldwide. There's roughly 500,000 new cases per day. 1.2 million deaths. 8,000 people are dying per day. And you notice that we are on a spike again. We are on an increase in the number of cases, and this is really the number of people who test positive, right? This is the number of people who test positive, but aren't necessarily sick. Not everyone who tests positive is sick, but if they test positive, they may be able to transmit the disease. To me, one of the more hopeful signs is that despite the rapid increase in the number of people testing positive, the actual number of deaths is remaining fairly steady. Now, make no mistake, it's tragic that anyone dies from this disease. But the fact is, as we talk about better treatments, fewer people are dying when they get the disease, even if they go into the hospital. To focus more in on the U.S., almost 10 million people in the United States have tested positive for COVID. We have 240,000 deaths, but look, 6.2 million people have recovered. All right. Now, Sanjay, John Hammond, the mathematicians out there, I'm going to pull a trick that I learned from my lab chiefs at NIH. They had their own special way of doing curve fitting, and I'm going to use that right now just to illustrate a point. So if we look at the number of daily cases in the US, this came from BBC, all right, what we can see is the number of daily cases is trending upwards. I'm sure you guys just love that quick and dirty curve fit method there. But the number of cases is definitely increasing. But if we look at the number of deaths, we spiked, and then they began to decrease, and now for the most part the number of deaths is remaining steady. And so that's why I drew these lines so that we can just sort of see the overall trend. Obviously we want to get to the point where there's 0 deaths, but 6 months ago, if our infectivity rate, if the number of positive cases was increasing at this rate, the number of deaths would also be increasing at this rate, but that's not the case now. So that's a little bit of a hopeful sign, in my opinion. If we begin to look at how COVID affects people based on race or ethnicity, what you see is that American Indians, blacks, and Hispanics have a much higher number of cases than Asian non-Hispanic people. Hospitalization rates are greatest for Native Americans, but it's pretty high for blacks and Hispanics, and the death rate tragically is highest for blacks. Okay. If we look at this on the basis of age, I want you to notice that here we're lower, right? Risk is lower, and here we are higher. So hospitalization is lower as long as you're under, 29 years of age, and then the older you get, the more likely you are to require hospitalization. And again, the older you get, the more likely you are to die if you get COVID. All right? Okay. Now, COVID do's and don'ts. How to become infected with COVID. Hang out in crowds. Go to the bar. Go to the restaurant. Right? Go to, sports events with lots of people. Go to political rallies. Go to protests where there's lots of people. Be in close contact with all those people and make lots of noise. Or sit in an enclosed space for a long period of time. Right? All of these factors contribute to your susceptibility to becoming infected with COVID. How to avoid being infected with COVID. Wear a face mask. Everybody has to wear a face mask. If you are wearing a face mask and the person next to you, the person across from you, is not wearing a face mask, you are doing a wonderful job of protecting them, because all those blue surgical masks, they protect other people from your respiratory droplets. But if the person across from you is not wearing a mask, basically they're just covering you with their respiratory droplets, and you are much more susceptible to disease. So everybody needs to wear those surgical masks, or you need to wear an N95 mask, which is very difficult to obtain these days. We want to maintain our social distancing, greater than 6 feet. Wash your hands often. Use sanitizer to clean surfaces. Quarantine your packages and things like that. All right, so we've been through all this. Everybody hopefully knows this by now. The problem is not everybody is following these guidelines. We're having COVID fatigue, so people are sort of going out and doing whatever they want, and that, I think, is part of the reason for this rapid increase in COVID again. Now, diagnostic tests. Back in May, we had very few diagnostic tests. Now we have at least 40 that have been attempted. All right, almost all these tests are PCR based, polymerase chain reaction, right? And all the PCR does is it just makes copies of the COVID RNA, and it makes enough copies so that you can detect its presence or absence. And it's very, very specific. One of the other nice things here is that back in May, we didn't have any FDA or European approved diagnostic tests for COVID, but now we have quite a few. So that's encouraging. I mean, think in 6 months, we've been able to cut through some of the regulations, but still maintain the quality. Otherwise, the FDA wouldn't approve of it. And now we have reliable tests. Okay. The other nice thing is we've cut the time down. Before, it used to take, 24 hours to maybe even 5 days to get a result. If you look here, some of these tests are able to generate a result in less than an hour. Certainly a number of them in less than a day, 4 to 6 hours. Okay. One thing that's beginning to appear, hasn't been successful yet, are home test kits. Notice all of these were discontinued because they didn't work. But rest assured that people are working on home test COVID kits. I know there's one supposedly available at CVS, but you take a sample and then you have to send it off for somebody else to actually do the test. I'm talking about home test kits that are like the home pregnancy test kit, where you can actually test yourself and get the result at home without having to go to the doctor's office. And so, that will save you from going to the doctor's office, potentially being exposed to other people, tell you that you need to watch your symptoms and so forth. So I'm going to guess that within 1 to 2 years at the latest, there will be some reliable at-home COVID-19 test kits. All right. The link over here is just where I picked up this graphic from. COVID treatments in use, right? If you have a mild case or mild symptoms of COVID, then essentially you treat it just like the cold or flu, but you keep an eye on yourself and know that these symptoms may last for 2 to 4 weeks, all right? If the severity of the symptoms increases, then you go see your doctor, and the doctor may prescribe certain therapies, one of which is dexamethasone or prednisone. These are steroids, and what steroids do is suppress your immune response. Specifically, they suppress your inflammatory response, because it's your inflammatory response that's actually causing all the damage that's making you sick. So the steroids knock that down, they suppress it. But steroids can also have neurological effects. They can affect your brain function. So if you take steroids, be sure that you follow the dosing regime exactly. Then the big headliner is remdesivir, right? This is the molecular structure of remdesivir. This is just a shout out to the chemists, especially the organic chemists out there. Remdesivir interferes with the RNA in the COVID-19. And then Regeneron has made an antibody cocktail, and it's a cocktail just because it's a mixture of 2 antibodies named Regen33 and Regen87. And basically companies just assign a number to each new drug that they make so they can keep track of it. So there were at least 87 attempts at making antibodies. These were the 2 that worked. And these 2 antibodies bind to, you guessed it, the spike protein on COVID, right? Regen33, Regen87 bind to the spike protein on COVID. So the spike protein is now covered up and it can no longer bind to the ACE2 receptor. Then that means that COVID can no longer infect the cells if these antibodies are binding it. So these are apparently very effective if they are given early on, before COVID has had a chance to infect your cell. Because once COVID is inside the cell, then it can replicate, make copies of itself. It gets released from the cell, and you're going to need a much, much higher dose of antibody to prevent those newly released COVID particles from infecting additional cells. So the sooner you get treated with this, the better. But antibodies are incredibly expensive to manufacture. So I really don't think, unless we find a cheap way to manufacture large quantities of antibodies, that this is going to be a last resort treatment. All right. The interesting thing, though, is Regeneron realized this, and the original dose of the Regeneron cocktail was 8 grams of antibody. Now that is a lot of protein to give to somebody, 8 grams. With clinical trials, they realized that they could reduce the amount from 8 grams to roughly 2 grams. So what we were dosing one person with 3 or 4 months ago, we can now dose 4 people with the full amount, because 8 divided by 4 is 2 grams per person, right? So that already is an improvement. This is, one of the points that I want to make, I hope to make during this talk, is just the amazing amount of rapid progress we have made in understanding, diagnosing, and combating COVID. There are also now treatment protocols in place, right? I stole this one from Boston Hospital. I'm not going to go through the whole protocol, but the idea is now there are actually algorithms, there are protocols that doctors can follow that have been proven to be effective. And again, this is what's reducing the number of deaths for people who have to enter the hospital with COVID. There are a number of therapeutic drugs now in clinical trials. So I've listed the drugs that are in clinical trials. The nucleoside analogs, notice remdesivir is one of them. Okay. but there's pegylated interferon, there's favipiravir, right? And I said that quickly so I didn't have to pronounce it correctly. But these interfere, the nucleoside analogs interfere with the COVID RNA. All right, so these are targeted at the RNA. Then we have interferons. These are small but cheap to produce proteins. Interferons were discovered in the 1950s, and they were named interferons because they interfere with the replication of viruses. But you have to be very careful because too high a dose of an interferon can cause lots of adverse events in the patient. Protease inhibitors have been tried because they were successful as anti-HIV drugs, because when COVID makes its protein, right, initially when it reads that RNA to make a protein, all of the proteins are connected one to another and they need to be chopped apart in order for the virus particle to form. The protease inhibitors inhibit or block those proteins from being chopped apart. So it gets stuck in that phase. Chloroquine we talked about in the spring. We think that may interfere either with the uncoating, the release of the RNA of the virus, or it may interfere with the actual release of the virus particle from the cell. And of course, as we all know, the jury is still out on the efficacy, the effectiveness of chloroquine. And then it does appear that antibody therapies work, but again, they're going to be probably prohibitively expensive to be used on a regular basis. But there are a lot of treatments. Again, this came from the same site, but there are a number of treatments here. And so they're classified partly by the target. What are they trying to interfere with? The stage is where they're at in the clinical trials. And we'll describe the clinical trial phases when we talk about vaccine development. And then the treatment goal, and what we notice here is that some of these treatments are actually not specifically targeted at COVID, but they're targeted at the symptoms that COVID causes, pneumonia or inflammation. And then I think it's important to note that this is a multinational effort to discover all of these different drugs. And so companies across the globe are working together. All right. Now, how do we end or at least minimize the COVID pandemic? And the catchphrase right now is this concept of herd immunity. And the idea behind herd immunity is if a high percentage of the population of us humans, we're the herd, all right, are immune to the transmissible disease. If most of us are immune to a transmissible disease, the disease will be limited in its ability to spread because there won't be any carriers. Every time the virus goes to infect a person, their immune system is already prepared to destroy the virus, so the virus transmission stops at that person who is immune. Vaccination increases herd immunity. The alternative is not to vaccinate and let everybody get sick, and that would just be a horrible solution. So we want to develop a vaccine. Unvaccinated individuals will still be susceptible, and for almost any vaccine, there's a small percentage of people who are unable to take the vaccine. They may be immunocompromised, their immune system may not work, but there's always going to be some people who can't take the vaccine. So the more of us who are vaccinated, we can protect those people who can't get the vaccine. The assumption is that at least 60 to 70% of the population must acquire some form of immunity, either by surviving exposure to the virus or hopefully by getting vaccinated before they get exposed to the real virus. Okay? The goal of a vaccine is to create an immune memory. Your immune system will remember the virus, and the next time you're exposed to that virus for real, your immune system will respond to it in hours instead of days or weeks. All right? And this graphic just illustrates what happens with and without immunity. So if we look at the top, the key here is the different types of people. So blue is somebody who has not been immunized, but they are still healthy, they're not sick. Orange is immunized and healthy, and red is they are not immunized and they are sick and they are able to transmit the virus. They are contagious. So in the first panel here, when no one is immunized and we have people who are in close contact with one another, then a very large percentage of people will become infected. And COVID does have a fairly high transmission and reproduction rate, all right? Now, when some of the population is immunized, this may, for example, be people who were previously exposed to COVID, got sick, and they survived it. Those people won't get sick, but there's not enough of them to protect the people who are not immunized. And so again, we have a fairly high percentage of the population who gets sick. But now look at this. If most of the population is immunized, when the virus tries to spread, basically the immunized people have presented a wall, a barrier to the virus from reaching the non-immunized people. Because when the virus interacts with these people, when these people are exposed to the virus, because they've been immunized, their immune system will eliminate the virus very, very quickly, and therefore these people won't transmit to the non-immunized people. So we have achieved herd immunity because the virus is very rarely transmitted now from one person to the next. And that's the purpose of creating the vaccine and of vaccinating everyone. So how exactly does the vaccine work? Yes, this is a Collins illustration. It's the best I could do with PowerPoint. So we're going to start off with our unvaccinated patient. They're not immunized. They do not have an immune memory. We've created the vaccine, and the vaccine contains inactive virus. So the vaccine contains virus or part of the virus that won't make you sick. It's inactivated. It can't make you sick, but it does have the viral proteins which your immune system can recognize. So here's our virus. We've isolated one or more of the virus particles, or we've just inactivated the entire virus. We can inactivate the virus by heating it or by exposing it to formaldehyde. Both of those things inactivate the virus, means it can't replicate, it can't infect you. All right? So we inject our patient with the vaccine. All right? The person doesn't get sick, but they do have some inactivated virus in them. And one of the tricks here with the vaccine testing is to make sure that we give them a high enough dose of these inactive virus particles so that their immune system will respond. So the immune system is activated, and lots of cells called B cells and T cells are produced, and they become memory B and T cells. The B cells produce antibodies, so your own body is going to produce antibodies which will recognize the spike protein on the virus. So your B cells will produce antibodies that recognize the spike protein on the virus, and we have tricked your body into making these B cells that make antibodies that recognize the virus by giving you the vaccine. So you didn't get sick, but you did activate your immune response to be able to recognize and attack the virus. The memory B and T cells now migrate to your lymph nodes all over your body, and they just hang out there until you're exposed to the virus for real. So they remember what the virus is. They're ready to protect you when you're exposed to the live disease-causing virus. And you guys notice that this was yellow, this was red. So it's the disease-causing virus, all right? You become infected, you're going to start to feel sick, just like if you get the flu vaccine. You may start to feel sick because the virus needs to interact with those memory B and T cells. And once the virus interacts with those memory B and T cells, those cells become activated. The activated memory and T cells begin to divide rapidly, right? And when that's happening, one of the things that you may notice is, we always touch our neck and say, oh, I'm getting sick. My lymph nodes are swollen. Well, your lymph nodes are swollen because your memory B and T cells are dividing and they're beginning to expand. And so your lymph nodes are getting bigger. They're swelling up. So when you say, oh, my lymph nodes are swelling, you should actually say, oh, awesome. My lymph nodes are swelling. That means my immune system is responding. I am making memory B and T cells. And those memory B and T cells, guess what? They're going to make antibodies that interact with the virus. They're going to bind to the virus. They're going to stop the virus from infecting your cells, so the amount of virus in you doesn't increase. And actually now with the antibodies attached to the virus, it makes it easier for your immune system to recognize and destroy the virus. So eventually you eliminate the virus. In the meantime, you've made more memory B and T cells, and they go back and live in your lymph nodes until you're exposed to the virus the next time. Now, one of the issues with the vaccine is how long will those memory B and T cells live in your lymph nodes, right? This is when we need to go and get revaccinated, right? So that's one of the challenges in discovering and producing a vaccine, is making a long-lasting vaccine. And people don't really know how long any of the COVID vaccine candidates are going to last. How long are those memory B and T cells going to live in your body? Is it going to be 3 months? Is it going to be 6 months? Is it going to be 10 years? So it may be that you may need to get revaccinated. You may need to get a booster vaccine every 6 months or every year as scientists perfect the vaccine. Quickly, this is a complex slide, so don't freak out here, but I just wanted to point out that one of the reasons that we've been able to achieve vaccine candidates so quickly is because we are now doing what we call rational vaccine design. And this incorporates bioinformatics and artificial intelligence. By interrogating the RNA sequence, by interrogating the proteins on the virus, we can very quickly come up with potential candidates, right? Antigens, things that stimulate your immune system. We can come up with candidates for the vaccine. And so we begin to test that out. We keep a database on that. And within 6 months to a year, and we've actually shortened that with COVID, but within 6 months to a year, we're able to create a vaccine that creates a number of pieces of proteins. These are known as epitopes, a number of pieces of protein, in this case, pieces of the spike protein on COVID that will trigger an immune response. And one of the first times that this rational vaccine was done was with the meningitis vaccine, because you'll see on the next slide, normally it takes 10 to 20 years to develop a vaccine. Once the DNA sequence of meningitis was discovered, it was 2 years until they had an effective vaccine. And now we're able to do this even faster. So vaccine components, what's in a vaccine? First of all, we have the immunogen. This is the molecule that actually activates your immune system. Then we have an adjuvant. This is a non-specific activator of the immune response. It just gives you a stronger immune response. The stronger your immune response, the more likely you are to get long-lasting memory B and memory T cells. But we don't want to overactivate your immune system. Otherwise, you get the same effects as if you had COVID. And then we have things called excipients, which stabilize the vaccine. These would be things like buffers that maintain the pH, right? There are a lot of different approaches to making vaccines. We talked about the whole inactivated virus. We have attenuated viruses. These are viruses that can replicate, but they have been manipulated so that hopefully they won't cause a disease in you. We can just give you the spike protein. Novavax, a company here in Montgomery County, is pioneering something called virus-like particles. So they've made an artificial structure that looks like a virus but isn't a virus. So it's going to be safer than either one of these two. And then we're looking at actually taking another virus and using it to deliver viral genetic material into your body. DNA, just giving you DNA, just giving you RNA. That's Moderna, which is on one of the next slides. And then a really expensive way is to train your immune cells ahead of time. Take immune cells out of your body, prime them with the virus, and then put them back in your body to activate your immune system. But this is going to be really expensive. So COVID-19, these are the key types of vaccines that people are working on. Live attenuated. So we're taking the virus and we're manipulating it so it can replicate but not make you sick. Inactivated, we've just killed the virus, but it still has all the proteins on the surface. Subunit, we're just going to inject you with one or more of the viral proteins, all right, or the DNA or RNA virus, and this is the Moderna approach. And so here is Moderna's messenger RNA vaccine approach. We're actually injecting you with RNA that contains the information to make the spike protein on the virus. So the virus RNA that makes just one protein, not the whole virus, just one protein, the spike protein, the information to make the spike protein is injected into your cells, and your cells take that viral RNA, they read it, Your own cells make the spike protein. They then release it into your blood or present it on their surface, and this triggers an immune response where we now form T cells, B cells, and other T cells, all the different types of T cells, and this will also then trigger the production of memory T and B cells, hopefully. All right. The advantage of this is an RNA vaccine is incredibly cheap to make. It should be very cheap. You can make it very quickly, and you can purify this to a very high degree, again, using fairly cheap and simple methods. To develop a vaccine, then, first we have to identify what the target is, the spike protein. Then we have to begin to synthesize that protein, and now we begin to test it. So we use animal models. We use mice. Ferrets are really good for the flu virus, and I think they're also being used for COVID. And then as a last resort, we use monkeys, macaques, but macaques are not very nice monkeys, and they are incredibly expensive to work with. Okay? Now, traditionally, right, the normal timeline for doing this was 5 to 10 years to do preclinical testing. All right? With COVID, we did this in really about 3 months. Said, yep, looks pretty safe, because really most of how we make COVID is the same way that we make the flu vaccine, which we've been making for over 50 years. All right? We then put this into people. In a phase 1 clinical trial, we're testing a small number of volunteers. Usually it's healthy male volunteers, and we do them in groups of 5, and we start with a small dose, and then we increase the dose, and we're making sure that nothing bad happens to those people when they get injected with the vaccine. And we're also testing to see if they make antibodies. In a phase 2, we go to a much higher number of people where there are going to be more genetic differences, and we may be able to pick up any adverse events, but we're still doing this at just one hospital. And then in a phase 3, we go to thousands of people, and we're going to do this at multiple different hospitals, because we're trying to see if the doctors or nurses who are administering the vaccine can get the same results that people did at the first single hospital center. All right. And again, so we've done this really in about a year. All right. We also have to plan for process development. How are we going to make large quantities of the vaccine? How are we going to ensure its quality, right? Quality control. And how are we going to formulate this? How are we going to put the adjuvant, the excipient, and the immunogen, the active ingredient, into the vaccine? And we also need to plan the clinical trials right as we do the preclinical testing. So if everything looks safe, we submit this to the FDA as an IND. The FDA reviews it. Then they approve the clinical trials. If the clinical trials work, we submit a BLA, a biological license agreement. The FDA reviews that. And if that looks good, then this goes on the market, and we do mass vaccination. And a phase 4 is when individual doctors report adverse events. And then the FDA decides if those adverse events are so much that we need to pull the drug from the market, or that the risk benefit is acceptable. How do we produce a vaccine? This is really one of the things that we teach in the biotechnology program here at Montgomery College. We start with cell culture because the viruses need to infect cells, so we don't actually do viruses in cells yet, but we do cell culture. The virus infects the cells because, remember, viruses can only replicate when they are in a live cell. We scale this up, and actually for biomanufacturing, it's not an individual flask. This goes into large-scale bioreactors, which will be 5,000 liters in volume. Right? Then we need to purify the virus from the cells and package it. We add our adjuvant, our preservatives, these are all excipients. Then we need to fill this into individual doses. We need to do quality control to make sure that we have the right dose, that there's nothing else in there that shouldn't be in there. And then we label it and we package it. Okay? This is just a little more detailed description of virus production. And I know that I've been talking a long time, so I'm going to just sort of skip through this. But one of the things that you need to think about with this is once we make the virus, we have to get it to the end user, right? So the factory makes the virus. We then have to deliver this. This usually goes to a distributor, a warehouse sort of place, right? The middleman. And then this is distributed regionally. and then this gets distributed to the doctor's office, to the pharmacies, and so forth. One of the critical aspects in biologics is the cold chain. A lot of times we need to maintain cold. This needs to be a plus 4 degrees C, which is refrigerator temperature, or the vaccine won't work. So we need to maintain a cold chain. Okay? And so, again, there are a lot of moving parts to this. And again, it just illustrates that biotechnology, that vaccine production is actually a multidisciplinary endeavor. And so, it's not as easy as it sounds, but the supply chain has already been planned. As soon as they thought they were going to have an effective vaccine, people began planning for the supply chain, even simple things like syringes. And that's one of the reasons that we're having a little bit of a shortage of flu vaccine, because there weren't enough syringes for both the flu vaccine and for the COVID vaccine. There are a number of COVID vaccines in clinical trials, and I will send a PDF of this out if you want to read more details. But the arrows here highlight local companies: AstraZeneca, Moderna, which is working with NIH and Lonza. These are companies that are here in Montgomery County. Novavax, GlaxoSmithKline is also working on 2 of these. And I'm also proud to say that each of these companies, Lonza, AstraZeneca, Novavax, and GlaxoSmithKline, has hired students who have completed the biotechnology program. So I think there is a lot of hope, all right? And a number of these vaccines are in clinical trials, and these actually started in mid-2020 instead of late 2020. Operation Warp Speed was $5 billion. A big chunk of that came to companies in Montgomery County, but $5 billion to develop a safe and effective vaccine. And it looks like, if everything holds true, that we may have accomplished that in just under a year, which is remarkable and really hopeful for all of us. All right. So this just illustrates the number of vaccines that are in development. There's lots of them, right? Lots of companies are making vaccines. First to market that's effective is going to make a lot of money. All right. So how close are we to a vaccine? As of September 8th, none are approved, but 9 are in phase 3. And remember, phase 3, if the vaccines successfully complete phase 3, then they go to the FDA, and the FDA can approve them. And we are expecting approval of several of these vaccines in late December, early January, maybe February. And all vaccines have a life cycle, and this is just another slide that sort of illustrates all of the different processes here. And I'm sure that your brains are fried by now. And so that is the end of this presentation. I hope that you found it useful. I hope that you guys stay safe, and thank you for your attention.